In this video, we'll talk about the pancreas, the liver, and the gallbladder. So before I jump into that, I want to just take a look at a picture and explain why we're taking these topics in this order. So far we've talked about the oral cavity, the mouth, the pharynx, the esophagus. We've spent, a decent amount of time talking about the structure of the stomach, the function of the stomach, the secretions, what's happening at the stomach. And then we also looked at the regulation of gastric secretion and the regulation of gastric emptying. So remember, after the food mixes with gastric juice, it creates this acidic paste called chyme. And the chyme exits the pyloric sphincter and it moves into the first region of the small intestine, which is called the duodenum. So the chyme is going to enter into this first region of the small intestine. Now this is the large intestine crossing over top of the small intestine. And so just so, we're clear, like this is where the chyme is headed. It's going to go through the small intestine, but we said that acidity within the duodenum is going to cause a sympathetic reflex called a gastroenteric reflex, and it's going to stop the stomach from emptying any further until that chyme is processed. So the way that the chyme gets processed in the small intestine has to do with the secretions of the liver, the gallbladder, and the pancreas. That's why I want to talk about those three organs next. And then just taking another look at a picture, different picture. So here they've cut everything away except the organs that we're going to talk about in this video. So here's the liver, here's the gallbladder, here's the pancreas, and this is the duodenum. So this is where the chyme is entering in. And I just want to point out that the liver, the gallbladder, and the pancreas, do you see how the secretions from all of those organs enter into the duodenum through one common duct? That duct is regulated by a sphincter. It's called the hepatopancreatic sphincter. So we'll talk about that, the regulation of allowing those secretions through. But the first thing that I'm going to talk about is the pancreas. Okay, because the pancreas produces some secretions, and they're exocrine secretions, and those secretions, let me just trace this out, will exit the pancreas through the pancreatic duct, okay, through this hepatopancreatic sphincter. But in the case that this sphincter is closed, take a look at this. Do you see how there's an accessory duct? And so the secretions of the pancreas can always go into the duodenum. But at any rate, that's why we're not going on to the small intestine right away. Let's talk about what the pancreatic secretions, liver, gallbladder secretions do in the duodenum. And then we'll pick it up in the next video with, the actual functions and secretions of the small intestine. So jumping back to our notes, in this video I'm going to start with the pancreas, and as I mentioned, it's an exocrine function with the digestive system, and that's the majority of the gland. 99% of the pancreas is exocrine in function. What we looked at in the endocrine system, the pancreatic islets, that makes up only about 1% of the pancreas. So this is an organ that's uniquely both endocrine and exocrine. But as far as the digestive system goes, we're just looking at the exocrine function. And so the hormones insulin and glucagon don't digest food. So they just help to regulate a nutrient level in the blood, as you know. So taking a look at the pancreas, we'll look at some more pictures from our book. Going back to this picture, this is a picture where you can see the pancreas. Here's the stomach, and then do you see this yellow structure peeking out kind of underneath the stomach? That's the pancreas. So the pancreas is not easy to see. Taking a look at this picture, we looked at this in the last video. You might recognize here's the stomach, and I don't see the pancreas. This is the lesser omentum, this is the greater omentum. I can see that was, severed. But the pancreas is up underneath the stomach. And so that's why, say like in this picture, I can see it just like peeking out there, just to put that in context. Because when you look at a picture like this, it makes it look like the pancreas is, just very obvious. It's not. It's tucked away. in this first C-shaped portion of the small intestine, and then the stomach is kind of hiding it, I guess you would say. Now, if we took a close look at the pancreas, here's another picture from our book, and they cut away the stomach from this picture. What they're doing here is they're showing us, you know, the cellular organization of the pancreas, and I'm just going to zoom in on that. So the pancreas is made out of these cells called acinar cells. Those are all of these, I guess, like pink cells, and these are exocrine cells. And so the makeup of the pancreas is a little bit like the lungs or like the mammary glands. It would be a good analogy here, because there's a branching duct system, and this branching duct goes into, these are called acini. They're just like little pouches or sacs, and this is where the secretion leaves, just like sweat or oil or salivary, you know, enzyme, something like that. And then that secretion would be conveyed, you know, through a duct out onto a surface. So here you can see the branching structure of the acinar cells. And that's separate from these pancreatic islets, the islets of Langerhans. So we're not talking about those. This unit, we're really concerned with the exocrine function. And I found this picture on the internet. I threw it in here. It's pretty nice because they did the outlining that I wanted to show you. So you can see kind of like the exocrine portion of the acinar cells, you know, versus the endocrine islets. So the pancreas secretes two different types of secretions. It secretes sodium bicarbonate, which is just a base, and it also secretes digestive enzymes. And the digestive enzymes that are secreted are digestive enzymes that will start to break down, chemically break down every category of food. Carbohydrate, sugar, protein, and fat. So the pancreas releases digestive enzymes that will break down all three categories of macronutrient. And that's the first time we're seeing that, right? Because the salivary glands released an enzyme that would break down sugar. The stomach released pepsin to break down protein. But the pancreas is releasing enzymes that will really start to work on all three of the nutrients that we consume. And so I'm going to jump back to the notes to show you how that works. Okay, so some of my other notes here. So the pancreatic cells are arranged into acini that connect to a duct system. We looked at that. And then the pancreatic duct connects to the duodenum at the hepatopancreatic sphincter. I showed you that, also known as the sphincter of Oddi. It's hormonally regulated as far as when it's opened and when it's closed. So we'll take a look at that. But we noticed that even if it's closed, the pancreas can still secrete its products into the duodenum. All right, and so what I did in the notes, just to try to make this easier, is I put everything into a table. But I think that I'm going to teach this looking at a picture, and then we'll look at the table, because the table is a summary of what's going on. So let's take a look at what's going on first, and then we'll go back to the table. So this is one area of the notes where I actually go into more detail than the book, just in one little part. I'll show you where. So what we can see so far is that here's the pyloric sphincter, and I can see here's the chyme. Okay, so that's the food mixed with the gastric juice, and I see that some of the chyme has moved into the duodenum, which we said then that's going to put the brakes on the stomach, a sympathetic reflex, because the small intestine is not suited for that acidity. So what will happen here is two different things, and I'm going to color code this on the picture. One thing that will happen is just the acidity. Okay, so the acidity of the chyme entering the duodenum is going to cause enteroendocrine cells in the duodenum to release a hormone called secretin. Okay, so secretin is a hormone. So basically what we're saying here is that the low pH, okay, just the acidity within the chyme, it's going to trigger some of these cells they call them in our book enteroendocrine cells. That's a very generic name. This is where I am a little bit more specific. So more specifically, these enteroendocrine cells are called S cells. And so these S cells release a hormone called secretin. And secretin is a hormone. It's not an exocrine product, and it's going to enter into the blood. So it's not going to enter into where the food is and mix with it. It's going to enter into the blood. And what secretin will do is when it reaches the pancreas, it's targeting the pancreas and some other areas too. We'll add this together in this video. But once secretin goes to the pancreas, what it does is it causes secretion of the sodium bicarbonate rich pancreatic juice, which makes sense, right? Because the bicarbonate is just a base. And so the purpose of the sodium bicarbonate that's coming from the pancreas is just to neutralize the acidic chyme. That's it. So it's just a pH adjustment, and that will happen. And that's the job of the pancreas, to neutralize that. The other thing that will happen is that just the presence of food, particularly fat and protein. Okay, so the presence of fats and proteins in the chyme trigger another type of enteroendocrine cell. I'm going to draw these purple. Okay, so these cells are scattered throughout the duodenum. These cells more specifically are called I cells. Okay, so the presence of fat and protein in the chyme are going to cause the I cells to release a different hormone called cholecystokinin. Okay, or people abbreviate that CCK because that's a long one. So cholecystokinin is a hormone. Just like secretin, it's going to enter into the blood. It's also going to target the pancreas, and it has additional targets. We're going to add those on in this video. But when cholecystokinin reaches the pancreas, this is a hormone that's going to cause the enzyme-rich pancreatic juice to be secreted. So remember, we said that enzymes that digest every category of food are released from the pancreas. And so the pancreas will release digestive enzymes as an exocrine product. I'm going to keep this color coordinated here. And it will also release bicarbonate as an exocrine product. The bicarbonate is just going to neutralize the chyme. And then the digestive enzymes, there's going to be a digestive enzyme for every single category of food. And I have that in the table. So I hope this makes sense. Basically, the acidic chyme, two different characteristics of it, will cause release of two different hormones from two different source cell types. Those hormones enter into the blood. Both of those hormones target the pancreas and cause secretion of the bicarbonate and the digestive enzymes. So taking a look at my table where I summarize this, I'll bring that a little bit back to original size here. Okay, so you can see in the chart, in the table, what I'm trying to do, I'm going to color coordinate this just like I did the picture. What I'm trying to do is I'm trying to show you that the pancreas is going to secrete two completely, different things. Just like the stomach, it gets secreted together. But unlike the stomach, they're regulated separately, meaning that the pancreas could secrete just sodium bicarbonate, right, if the hormone secretin was present only, or the pancreas could only secrete digestive enzymes if only the hormone cholecystokinin was present. But we have these two different types of secretions coming from the pancreas. An inorganic substance, which is sodium bicarbonate. That's the structure of sodium bicarbonate. Okay, so in words, you could just write out sodium bicarbonate if that helps. And the only job of the sodium bicarbonate is to neutralize the acidic chyme. So the chyme is going to come in there very acidic. It's going to come in at like a pH of 1. And sodium bicarbonate is going to be secreted from the pancreas until that pH goes up to around neutral, pH of 7. At that point, there would be no more acidity to make the S cells release secretin. And so the stimulus for the sodium bicarbonate secretion stops. So as far as regulation of secretion, it's the acid in the acidity of the chyme that will make cells called S cells that are within the small intestinal wall release a hormone, secretin, and secretin is going to have the effect of stimulating release of bicarbonate from the pancreas. So I hope this makes sense that as the pH starts to increase, then less secretin is put into the blood. And so at a pH of 1, we're, putting sodium bicarbonate into the small intestine quickly. But as that pH comes up to 3, 4, not as much stimulus, sodium bicarbonate is more slow. At a pH of 7, I want to stop sodium bicarbonate secretion because if you keep secreting a base, it's going to go up to 8, 9, right? So it will become basic, and we don't want that to happen either. So that is that part. The other part is this digestive enzyme mix. So all these digestive enzymes are secreted together. There's not separate regulation for them. Okay, so all 3 digestive enzymes are secreted together. The digestive enzyme that secretes, that digests sugar is amylase, and we just say pancreatic amylase because it's the same as the salivary amylase. It's just coming from the pancreas. And more specifically, what it does is it takes a complex sugar, a large branching chain, and it cuts it into dimers, or like just pairs of sugars, so 2 monosaccharides bound together. There is an enzyme called pancreatic lipase, and we can tell by the name of that. That's going to break down fats. It breaks down triglycerides into fatty acids and monoglycerides. And then we're also going to digest proteins. And there are 3 protein digesting enzymes that are released. Of course, just like we talked about in the previous video, they're released in their inactive form. Do you remember what that's called? A zymogen. Good, zymogen. So I listed the zymogens here. So trypsinogen, chymotrypsinogen, and procarboxypeptidase. Those are the 3 zymogens that are released from the pancreas, and all 3 of these digest protein. So these must be activated in order to digest protein into amino acids and small peptides. So I'm going to talk about how that activation occurs in a minute. I'm just going to go over to, how did we get this mixture of all 3 of these, categories of digestive enzymes. Remember, it was the presence of fat and the presence of protein in particular that made eye cells in the small intestinal wall release a hormone, cholecystokinin. And cholecystokinin targets the pancreas, and it stimulates release of all of the digestive enzymes from the pancreas. So, as I said, the interesting thing about these 2 categories of secretions is that they're regulated separately. And so, for instance, what if the contents of the small intestine were at a pH of 7, but there's still nutrients that need to be digested? I don't want these 2 things to be tied together, because if I had to keep secreting digestive enzymes with the sodium bicarbonate, as we said, that pH is now going out of range in the other direction. It's going up to 8 or 9. Or, what if all of the food was completely digested, yet it was still at a pH of 4? I would still need to neutralize that. So I wouldn't want this ability to neutralize the contents to be turned off when the digestive enzymes get turned off necessarily. So they're, they can work independently, and not only can they, these 2 categories be secreted independently, but just jumping to a picture here, we'll use this picture. But remember, the hepatopancreatic sphincter is regulating secretion, but even if that wasn't open, that sodium bicarbonate and digestive enzymes can always get in to the small intestine through that accessory duct. Okay, so going back to our notes, let's take a closer look at the protein digesting enzymes, and then we'll move on. So the pancreas releases protein digesting enzymes. We said that there are 3 of those, and one of them, trypsinogen, is activated by an enzyme that's embedded right in the wall of the small intestine. It's called entropeptidase. So in other words, it doesn't even take a condition or, another secretion. The enzyme is right there. All you have to do is get this enzyme out of the pancreas, and in the wall of the small intestine, this entropeptidase will convert the trypsinogen into trypsin. Okay, so entropeptidase does that activation. Once we have trypsin activated, it's trypsin that converts the other two zymogens, and it's going to convert the chymotrypsinogen into chymotrypsin. That's a protein digesting enzyme. And it's going to convert procarboxypeptidase, the zymogen, into the enzyme carboxypeptidase. Okay, so the three protein digesting enzymes in their active form are trypsin, chymotrypsin, and carboxypeptidase. All three of those will digest protein in the small intestine. And so just taking a look at a few pictures to support this. This is a picture from our book. This is showing us secretion of digestive enzymes, but you see how they're really kind of focused on what we were just talking about. They're saying that there is this membrane-bound enzyme, entropeptidase, that when trypsinogen, chymotrypsinogen, procarboxypeptidase, that's a mouthful, are released, that the trypsinogen first gets converted to trypsin, right, by the entropeptidase. And then with this arrow, they're trying to show us that the trypsin is then converting the other two into their active forms. Now you might wonder, why do we need so many protein digesting enzymes? If you think about protein digestion, there was no protein digestion in the mouth, right? That was just amylase, that's sugar. No digestive enzyme secreted in the pharynx, none secreted in the esophagus, pepsin in the stomach. And now we have trypsin, chymotrypsin, and carboxypeptidase. Why do we need 4 protein digesting enzymes? And I just want to talk about that a little bit, because these protein digesting enzymes, what they do is they recognize certain combinations of amino acids and clip it right at that spot. So if you had a when we ingest a protein, a full protein, hundreds of amino acids, at first pepsin is going to work on that. It's going to cut it into smaller pieces. So now here we have polypeptides, smaller pieces of protein. And then what trypsin and chymotrypsin we could add to this. I'm not going to write that out, but the trypsin and chymotrypsin, they will take these peptides that were elicited by the pepsin and break them down even further, like I said, by cleaving at a specific junction of 2 amino acids into these little peptides. Peptidases, which we haven't talked about yet, and the carboxypeptidase, remember that comes from the pancreas, that one works a little bit different. That one is not looking for a particular, combination of 2 neighboring amino acids and, cutting at that point. What carboxypeptidase does is, remember, all of these, the protein is made out of amino acids. Amino acids have that NH2 group, the amine group at one end, and the COOH, the carboxylic acid group at the other end. I just grabbed these pictures from chapter 2 just so we would remember this. So here's a short peptide, and so with the structure of the amino acid in mind, does everybody see here's the amine group? It's free on one end of this peptide, and then this is cutting off a little bit, but there's our carboxylic acid group on the other end of the peptide. Carboxypeptidase, carboxylic acid, this is the only enzyme that can actually take a peptide and start just cutting individual amino acids. So what the carboxypeptidase does is it cleaves at the carboxyl acid end. So it would cut off this amino acid, and then you'd have this carboxyl group, then it would cut off the next amino acid. So it works on that carboxyl end of a peptide to elicit free amino acids. The other ones, pepsin, trypsin, chymotrypsin, what they do is they basically, if you want to think of it this way, just chop them up. So I get little pieces like this. I don't get the individual amino acids, but the carboxypeptidase will do that. So I hope that makes sense. That is the pancreas. So let's move on to the liver. So the liver is a vital organ. The pancreas is also a vital organ. We need both of these organs to be working to sustain life. And to understand the function of the liver, it helps a lot to understand the macroscopic and microscopic anatomy. So I'm going to switch to some pictures, and then we can come back and mark this up. Okay, so here's a cadaver shot of the liver. The liver is organized into lobes, just like the lungs. Okay, so there's a major lobe over here. This is called the right lobe, and this is another major lobe. This is the left lobe. This is the falciform ligament that anchors the lobes, and it also attaches to the diaphragm. And then this little thing peeking out here is the gallbladder. So the liver is organized into two major lobes, which you can see, and then there are two minor lobes. The two minor lobes are on the inferior surface of the liver. So here we're looking at the bottom of the liver. And I can tell this is the back because this is the inferior vena cava. Okay, so does everybody see that big, collapsed blood vessel there? This is the gallbladder. That's another reason I can tell this is anterior. And right next to the gallbladder, there's this little segment. This is a minor lobe. That's the quadrate lobe. And back by the blood vessel, this little segment, that's a minor lobe. That's called the caudate lobe. So there's the caudate and the quadrate lobes. Those are minor lobes of the liver. So we have two major lobes, two minor lobes. Regardless, each of the lobes is organized in the same way. And I'm going to take a look at that. So in our book, what they're doing is they're showing us a section, a microscopic section through the liver, because now we're starting to talk about like the microscopic structure, like how are the cells arranged within the liver. And they're arranged, each lobe is organized into lobules. So you can see this really very neatly arranged pattern of cells. Here they're zeroed in on, this is a lobule. And we can see this blood vessel, or at least where the blood vessel was on the inside, that's a central vein. And so the way that the lobule is arranged, and I'll show you some pictures, is that the hepatocytes, or the liver cells, are kind of organized in plates. And those plates radiate out from this central vein, kind of like spokes on a wheel. Okay, so that would just be, plates of these hepatocytes that we're seeing, and I'm just kind of conceptually showing you how that works. So the hepatocytes kind of make up this lobule by radiating out from around that central vein. Now, the way that the liver works basically is the liver processes our blood. It filters items out of the blood, it metabolizes all of the nutrients that are there, including metabolizing things that aren't nutrients like alcohol, medications. We would call that detoxification. The liver takes dead red blood cells out of circulation. We've talked about that before. So there's a lot of jobs of the liver where it's basically processing our blood. And so see if this makes sense. I want to try to explain this just very generally. So the way that a lobule works is the blood that enters into the lobule is going to enter in from the outer periphery. From many, many different areas, but I'm just drawing one area. So the blood would actually come in from the outer periphery, and it flows towards that central vein, which makes sense. And by the time the blood reaches the central vein, the work of the liver is done. The work of the liver gets done because the blood is flowing through this really leaky capillary. It's called a hepatic sinusoid. You probably remember the liver contains sinusoids, which are the very porous, leaky capillaries. And on either side of the sinusoid are the hepatocytes. So what happens as the blood flows to the central vein is that these hepatocytes, actually process the blood that's flowing by. So that by the time the blood hits the central vein, the work of the liver is done, and that blood is going to exit the liver. So that's a basic description. Now let's take a look at a few pictures and see if this makes sense. So here is a diagram from the book, and what we can see here is a single lobule. Okay, so here's the central vein. You can see in pink the hepatocytes that I mentioned, how they radiate out, kind of like the spokes of a wheel, from that central vein. And then I can see blood vessels on the outer periphery. We know the blood is going to come from the outer periphery and flow to the central vein. So the blood comes from two different sources. We're going to have fresh, clean, oxygenated blood coming in from the hepatic artery. Okay, so that is a branch off of the descending aorta. And then we also have blood coming in from, this is the hepatic portal vein. So this is part of a portal system that we haven't talked about yet. Now, this is the first time I'm talking about it. Remember that a portal system is when you have two capillary beds, one right after another, like we saw surrounding the hypothalamus and then around the anterior pituitary. So the hepatic portal vein is bringing blood that's already flowed through the capillary beds of the small intestine. So think about that. And we'll talk about this again when we get to the small intestine, but the blood that's flowing through the small intestine, that's the blood that's going to receive the absorbed nutrients. It probably might also receive other things that we didn't mean to ingest, things that might be harmful to us, right? And so you don't want that blood that is picking up all of these items from ingestion just being circulated through the whole body. So the blood from the small intestine then gets shuttled to the liver. That's what you call the hepatic portal system. And so that's the other source of blood, this blue vessel, because it's, already gone through the small intestine, but this is the hepatic portal vein. So blood that has flowed through the small intestine that might have a lot of nutrients in it, which the liver will process, also goes to the liver. So the fresh oxygenated blood, the hepatic portal vein blood, this blood is going to flow through. This is the hepatic sinusoid. And you see how as that blood passes through, these hepatocytes on either side are going to metabolize the nutrients that are there, metabolize the other substances that are here, take dead red blood cells out of circulation. That's a specialized cell type that's here. It's called the Kupffer cell. It's a type of macrophage. They're specialized to take the dead red blood cells out of circulation. So it's doing its work, and then by the time that blood hits the central vein, now the blood's going to leave the liver. So let's look at that story one more time. This is a picture from a different book, but it's the same idea. Do you see how as long as you know how this works, doesn't matter how it's depicted. Here's the hepatic artery. Here's the hepatic portal vein. And blood from both of those areas are going to move in towards the central vein. And as that blood passes the hepatocytes do their job. And in this picture, they're even showing us these pink cells, the Kupffer cells. So those are the ones that are specialized for taking the dead red blood cells out of circulation. So I hope that all makes sense. Let's go back to the notes and take a look at what we have here. So macroscopically, there's 4 lobes. There are 2 major lobes, 2 minor lobes. The 2 minor lobes are the caudate and quadrate, and the 2 major lobes, right and left, we looked at those. Each lobe consists of subunits called lobules. And so as far as what is a lobule, this is microscopic. We can't see this with our bare eyes. Basically, it's hepatocytes, the liver cells and Kupffer cells that radiate out from around a central vein. And so blood flows into a lobule from the outer periphery, past the planes of hepatocytes in a vessel called the hepatic sinusoid to the central vein. Blood flowing through the lobule is coming from 2 sources, the hepatic artery and the hepatic portal vein, bringing blood that's being shuttled into the liver from the small intestine. So as the blood passes through the hepatic sinusoid, what is the liver doing? We said it's a great processor of the blood, and it will metabolize every category of food. So it filters dead red blood cells, for one thing, out of the blood. It processes nutrient-rich blood, so sugar, protein, and fats will be metabolized. Fats are actually removed by the liver, and we use it to make something called bile. Some of those fats we'll use to make bile. Store fat-soluble vitamins. Iron would be stored by the liver. detoxification I mentioned, that would just be the metabolism of substances other than like a nutrient source, like I was saying, like a medication. But then other things that the liver is doing, remember we talked about this before, the liver produces proteins. Over 90% of the proteins that are found in plasma are produced by the hepatocytes. And it's that real leaky vessel, the hepatic sinusoid. And so items like albumin, angiotensinogen are produced by the liver and then put into the hepatic sinusoid. That's how they make it into the blood. And then the liver is also producing bile. And so I want to talk about that for a minute and see if this makes sense. I'm going to jump back to one of my pictures here. I'll look at this one from our book. So as blood goes from the outer periphery into the central vein, and we're metabolizing what's there and taking out dead red blood cells, you can imagine that we're taking out items from the blood that we want to remove from the blood. We're cleaning the blood, right? So what does the hepatocyte do, with this waste? What do you do with the dead red blood cells and, the other metabolites? You can think of it this way. All of these hepatocytes have access to what are called bile canaliculi. You can see this kind of like green, looks like a web that they're pulling up here, but really it's just a series of channels. And so you can think of it as kind of like the garbage dump. So the hepatocyte takes the garbage out of the blood, and then it puts that garbage into the bile canaliculi. Okay, so bile canal. And so basically what bile is an excretory product. It's literally just, how are you going to get rid of the waste that was collected by the liver? Well, the liver will produce a product called bile. It's very similar to the kidney. You see how the kidney wants to remove waste from the blood. What are you going to do? You produce a product called urine. And so bile is an excretory product. It does have a few other roles, which we'll talk about, but basically it's an excretory product. So taking a look back at our notes, the production of bile has a lot to do with just getting rid of what we don't want. So some of the main elements of bile are parts of the dead red blood cell, so bile pigments. Remember that the Kupffer cells will phagocytize the dead, damaged red blood cells. And what they do is they break down the globin portion into amino acids. And then that part's easy. And then they metabolize the heme group by extracting the iron and storing that. And then that organic frame that was around the iron gets metabolized into bilirubin. So that is a bile pigment. And just to look at that, look at a picture quickly. We've looked at this so many times, but just to remember this. So here's hemoglobin. Remember that every red blood cell contains millions of molecules of hemoglobin, like hundreds of millions of molecules of hemoglobin. And so we have these 4 protein chains. We know that these are just made out of amino acids. That's easy to metabolize. We know how to metabolize a protein. This heme group presents a little bit of a challenge because we need to extract the iron and save that to make brand new hemoglobin, which the liver does. Like we said, it will extract the iron, bind it to a carrier so that it's safe in the body and reuse it. But this is the part that turns into bilirubin. Okay, so that organic ring, we don't reuse that. It is a pigment in red blood cells, as you know, that when oxygen binds to this iron, it appears more red. When oxygen is not bound to the iron, it appears more blue-green. Here, oxygen is not bound. This is a dead red blood cell. And so when you metabolize that ring, it still has that pigment quality. And it does give bile its green color. Okay, so the bile pigments are just that portion of the dead red blood cells. Bile salts are fats, basically. They're made out of cholesterol, and the liver does produce cholesterol and put cholesterol into the bile and produce cholesterol for the body. But they're amphipathic molecules, which means that they're partially hydrophilic, partially hydrophobic. And what they do is they emulsify fat in the small intestine. And what that does is it increases the surface area. And so I'm going to talk about the emulsification of fats in my next video too. But basically what a bile salt will do is it will take the fat that's in our small intestine, because inside of the small intestine is going to be a watery environment. So even if I take, a bite out of a stick of butter and I chew it into, 100 pieces, by the time it makes it down to the small intestine, we know that fat is going to aggregate together in a watery solution. So it doesn't matter how much I chewed it up, the fat aggregates back together. In order to digest this, I need that fat to get broken down into small droplets of fat, because then the fat digesting enzymes, or the pancreatic lipase, can work around the outer periphery of these droplets. And so this is what the bile salts do. They don't digest the fat. Okay, lipase will do that, but what the bile salts will do is take the fat and disperse it into small droplets. That's what's known as emulsification. So that's what I mean there by emulsify the fat. And then bile also contains water and electrolytes. And so because of that, it works somewhat like a buffer, to stabilize the pH. But it's getting rid of that dead red blood cell waste. And also the bile salts will emulsify the fats. And so those are the main functions of bile. And if it makes sense to you, the story that I told about the liver, how it's just continually, filtering the blood, metabolizing the nutrients, making proteins and so on. The liver's also 24/7 making bile. Kind of like the kidneys, sounds like a very similar story. But like the kidneys, we're not going to continually release the urine, right? There's going to be a storage place for it, the urinary bladder. And for the liver, there's another storage place called the gallbladder. The gallbladder is just a muscular sac that will store this bile until it's ready to be secreted in the small intestine. So I'm going to change pictures here. This is a picture we'll look at again in the next video. This is the description in the book that I just tried to give, that in the small intestine, all of our dietary fat will aggregate back into one glob, and so bile salts will help to emulsify it so that digestive enzymes can break it down. So that's that picture. And then I wanted to take a closer look at this duct system and see how bile is going to be secreted and or stored. And I put a version of this picture in the notes. So I'm going to jump over the notes because we're going to be able to mark that up together. Okay, so here's the picture that I'm referring to. See if this makes sense. So the liver was removed from this picture, so we'd still have the liver, great big organ, right in here. And so when bile is produced, the way that it leaves the liver is through these right and left hepatic ducts. Okay, they're going to take the bile out of those two major lobes, and where that leaves the liver, that's called the common hepatic duct. And that bile is going to go down the common hepatic duct, and once it passes where the gallbladder ties in, it's known as the common bile duct. Okay, so here they're just saying bile duct. So that's the common bile duct. So we'd have the common hepatic duct, and then, like I'm trying to describe, once the gallbladder duct ties in, which I'll name that in a minute, then we call this the common bile duct. Okay, and it's the common bile duct that comes down. It's regulated by the hepatopancreatic sphincter. So the way that this works, I'm going to change my marker color here, maybe make it easier to see. I'm going to make the bile yellow, is that bile is continually being produced. And that bile is going to continually go down this path right up to the hepatopancreatic sphincter. So hepatopancreatic sphincter is usually closed, right? It's going to be opened hormonally, and I'm going to talk about that in a minute, how that works. And so when it's closed, what will happen is that bile is going to back up the common bile duct. And when it gets to this point, because we have bile traveling down from the liver in the common hepatic duct, it's going to take the path of least resistance. It's going to take this side road. This is called the cystic duct. And the cystic duct is the duct that leads into the gallbladder. So the gallbladder, as I was mentioning, it's just a muscular sac where bile is stored. Unlike the urinary bladder, which I was describing how the urinary bladder doesn't change the composition of urine. Same thing with the gallbladder. I mean, it's just storage, but here's the thing. It could be stored for a long time, and as storage occurs, water will continue to be reabsorbed from the bile. So we'll keep maybe concentrating the bile within the gallbladder, if that makes sense, which can lead to the formation of gallstones, if you've ever heard of that. So a gallstone is precipitate that's forming in the bile. And it makes sense because water continually, is reabsorbed from the gallbladder. So when this hepatopancreatic sphincter is opened, what will happen is bile that's being produced by the liver can empty in, but the gallbladder will also contract and eject all of the bile that was stored up until that point. The gallbladder, as you know, it's a non-vital organ. We can live without the gallbladder. Many people do. And all that will happen is the bile is going to back up in this duct system. Okay, so we don't actually need a specialized sac to store it. And people with their gallbladder removed will acclimate to that. So let's take a look at this picture. This picture talks about the hormonal regulation of bile secretion. And it's tied into the two hormones that we already learned. And so let's mark this up. Remember we said that when the chyme moves from the stomach into the small intestine, that it would cause release of secretin and cholecystokinin. So that's still the same story. So that low pH is going to cause the hormone secretin to be released from S cells. That is a hormone. It's going to enter into the blood. And then the other part of this story was that the fats, right, and protein that was out here, I'll just put P for the protein, those nutrients. So acidic, fatty, chyme. Oh, technically, keep this separated, even though the book's not separating. The acidic portion causes the release of the hormone secretin, and then the fatty portion, particularly fatty, will stimulate release of cholecystokinin. So hopefully you still remember that. Number two, those hormones enter in the blood, right? They're not exocrine. Nothing's happening out here with where the food is. Those hormones have an effect at the level of the pancreas, which we talked about. So that's still true. But right now I'm not focused on that. So I wanted to show you that what they do as far as the liver and the gallbladder are concerned. So what secretin will do is secretin stimulates the liver to produce bile more rapidly. Okay, so secretin has an effect also at the liver, and it stimulates bile production. Also, remember the bile salts that were part of bile that will be released into the small intestine. Once those bile salts emulsify fats, they get absorbed into the blood, and the bile salts get recycled back to the liver. I'm going to talk about that again in the next video. I know that's a quick explanation there. But the cholecystokinin, that is going to have its effect at the gallbladder. It causes the gallbladder to contract, and it causes the hepatopancreatic sphincter to relax so that bile will enter the small intestine. And I put this in a table too. Okay, so two hormones. Cholecystokinin is going to be released from the eye cells in the small intestine due to the presence of fat and peptides. And it targets the gallbladder, causes contraction of the gallbladder, ejection of the bile, and it also targets the hepatopancreatic sphincter, causes it to open. The other hormone, secretin, comes from mast cells in the small intestine. It's secreted due to the acidity. Secretin is going to target the liver and stimulate bile production and secretion from the liver. So that is the basic story of this video. So to finish up the notes, bile circulation, I mentioned this in the picture. Bile salts, because they're released into the bile, and what they do is once they're released into the bile, they emulsify the fat that's in the lumen of the small intestine. Those bile salts are absorbed into the blood like a nutrient. And so the bile salts, after they do their job, they get absorbed back into the blood. And remember, the blood is going to enter the hepatic portal system. So the hepatic portal system delivers this blood to the liver. Okay, so that's the second capillary bed. And bile salts are removed by the hepatocytes, and then they get re-added to the forming bile. Okay, so the bile salts are recycled, and when bile salts are coming back to the liver, it's an indicator that we're in a fed state, and it stimulates the liver to produce more bile, and you're giving it some of the ingredients of bile. Gallbladder, we know that this is non-vital, small muscular pouch tucked into the liver. All it does is it stores bile and it concentrates bile while it's there. And I wrote out the duct path that we labeled on this picture. And that is coming out of the liver, right and left hepatic ducts convey bile out of the liver. And then they merge to form the common hepatic duct. That common hepatic duct is going to change names to the common bile duct once the gallbladder joins. Common bile duct attaches to the duodenum. The opening of the duct regulated by the hepatopancreatic sphincter. And quiz question here, do you remember what hormone opens the hepatopancreatic sphincter? Cholecystokinin. I bet you got that. So if the sphincter is closed, what will happen is just the bile backs up in that duct system. And that's how it gets into the gallbladder in the first place for storage. So I will be back in another video to talk about the small intestine and the large intestine. Thanks.